Fairfax Cryobank, the trusted choice for donor sperm. This DVD is intended to provide information on our shipments, how they should be handled, and specifics about our thaw instructions. We encourage your lab staff to be in touch with us if questions arise. We would like to tell you about how our specimens should be handled when they arrive at your office, and what you'll find when you open the shipping tanks that contain the semen vials. This information can be found in our instructions for semen identification, thawing, and product specimen quality standard located on our website. Specimens are shipped in tanks that maintain temperatures below -130 degrees Celsius. Exposure to temperatures above this will compromise the samples. It is very important to know that any specimens exposed to room temperature for more than 3 to 5 seconds will be compromised. So plan ahead when examining or removing specimens from our shipping tank or your storage tank. When the shipper is received, it will be sealed with a zip tie for the safety of the samples and those handling the tank. The staff will carefully cut the zip tie, quickly remove the vials, and either transfer them to a storage tank or prepare them to be used for the insemination. The tank will contain the following. Summary of records. The summary of records is a document that records the information of the relevant infectious disease testing that is associated with the samples. This is an FDA requirement. Packing slip. The packing slip contains information such as the patient name, donor number, number of vials shipped, specimen prep, and expected post-thaw analysis numbers. These are our laboratory's analysis of the expected number of motile sperm per milliliter in that specific specimen. Fairfax Cryobank has a specimen quality standard that applies at the time the specimen is thawed and before any processing or washing. Sperm counts will vary 10 to 30% depending on the lab personnel and count method. Fairfax Cryobank's specimen quality standard states that IUI and ICI samples will have 10 million motile cells per vial, and IVF samples will have 5 million motile cells per vial. It is important to note that the specimen quality standard applies to the samples analysis prior to any additional processing or washing that your clinic may do. The staff should use cryo gloves to remove the sample from the shipper. Check the packing slip to verify the identifying information. Move the samples to a storage tank immediately, or proceed with the thaw procedure. There are 2 possible thaw procedures. You will find the determination of which procedure to use on the packing slip. Please follow the thaw procedure as directed, or you may compromise the quality of the sample. Always keep the vial in the upright position while thawing. Place the frozen vial in a thaw block at 37 degrees Celsius for 10 minutes, or until completely thawed. Remove from the thaw block promptly and begin your analysis. Once the specimen is completely thawed, wipe any condensation from the outside of the cryo vial and then unscrew the cap. Gently but thoroughly mix each specimen using a vortex or by pipetting up and down using a 200 microliter pipette tip. Perform an evaluation before any additional processing. Place 10 microliters of the recently mixed specimen on a microscope slide. Cover with a cover slip. Place on a 36 degree centigrade slide and allow to equilibrate for 5 minutes. Determine total motility and follow individual lab protocol using the preferred counting chambers to determine total sperm concentration. Finally, re-verify the specimen identification, including donor number and patient name prior to insemination. Note: if your clinic does not have a thaw block, the following alternates may be used. Please be aware that suboptimal results may be achieved. Alternate 1. Thaw in a 37° centigrade incubator. Remove promptly from the incubator after thawing. Or alternate 2. Thaw at room temperature on the countertop for 15 to 20 minutes, or until completely thawed. Please note, using a water bath is never acceptable for thawing a sample. If your specimen is in a straw, stand the straw vertically on the tabletop at room temperature for 10 to 15 minutes, or until completely thawed. Once the specimen is thawed, wipe any condensation from the outside of the straw. Open the straw by cutting one end, and then inverting the straw so that the open end is in a sterile container. Cut the other end and let gravity drain the contents of the straw into the sterile container. Gently but thoroughly mix each specimen in its respective sterile container by pipetting up and down using a 200 microliter pipette tip. To perform a post-thaw evaluation, place 10 microliters of the recently mixed specimen on a microscope. Cover with a cover slip. Place on a 36° slide warmer and allow to equilibrate for 5 minutes before analysis. Re-verify the specimen identification, including donor number and patient name, prior to insemination. If your sample is in a vial, please see the original processing paperwork enclosed for thaw instructions from the laboratory that froze the sample. Once again, re-verify the specimen identification, including donor number and patient name, prior to insemination.